Hello, everyone. My name is Thea Patil, and I'm a junior at Lambert High School in Suwanee, Georgia. My research was on a type of cancer called DLBCL, and in my research, I analyzed the genetic predispositions and environmental factors leading to causations of this cancer. So what exactly is DLBCL? DLBCL stands for Diffuse Large B-Cell Lymphoma, which is a type of non-Hodgkin's lymphoma. And B cells are a type of lymphocyte or white blood cell, which assist the immune system in fighting infection throughout the body. These white blood cells travel all throughout the body through a system of tubes and can access every organ. And the B cells work alongside T cells, which are another type of white blood cell, but in this type of cancer, the T cells are not affected. However, when a cancerous B cell is produced and starts replicating, the cancer cells are non-localized, meaning that they spread all throughout the body, which is why it's called diffused. It's a very fast-spreading and very dangerous cancer, which is why research is even more necessary to make sure more people have a higher survival rate for it. In my research, I tried to answer the question, how do genetic predispositions and environmental factors impact the onset of DLBCL? You can go to the next slide. So in my literary review, I focused on potential genetic and environmental causes of DLBCL, and I also wrote about some current and future treatments for it. So for genetic abnormalities causing DLBCL, I found that mutations, especially in specific areas of genetic code, create higher chances of cancer development. So for example, there's a very famous gene called P53 that acts as a tumor suppressor, meaning that it regulates cell division and prevents cancer cells from replicating. And this gene is at work all the time in every one of us. It's not only active in just people who have cancer. When the P53 gene gets mutated, it can have catastrophic effects and it can cause cancers like DLBCL. Chemical exposures can also cause changes in the body, including those genetic abnormalities that increase the chances of cancer. So for example, there's a chemical called polychlorinated biphenyls or PCBs, which are found in old paint and water pipes. And when we ingest these or inhale these, it accumulates over the years in human fats and lipids because it is lipid soluble, and it will not leave the body through waste because it will not dissolve in liquid. So over years, these chemicals build up in the fat tissues and cause genetic abnormalities leading to cancer like DLBCL. In other chemicals like pesticides, insecticides, fertilizers, and other agricultural products also have effects on respiratory and digestive health of humans, which can lead to lung and colon cancer. And there are other chemicals such as dioxins, some aerosols, and glyphosates, which are all found in very common household items that have the same effect. And lastly, it may be surprising to hear that it is supposed that cow milk can also cause DLBCL and other types of non-Hodgkin's lymphoma. So when we drink bovine milk, we also consume the cow's microRNAs, which interfere with the human IGF-1 signaling pathway. And this pathway is active during puberty, and it can also have an effect in preventing breast cancer. So if this pathway is disrupted, it can lead to childhood cancers like leukemia and breast cancer. When the microRNA interferes with the pathways, it prevents cell apoptosis, which is cell death. And this is bad because if a cell is cancerous, the body cannot kill it and it will continue to replicate into a worse cancer. Other food items, such as those containing the red 40 food colorant, are also carcinogenic. So that's in a lot of different foods, and we have identified that to be carcinogenic. So for my methodology and results, I identified that for genetic risk, individuals at a high risk for cancer in their family should genetically test themselves for mutation markers that are commonly identified with cancer. So there are specific sequences of gene code that can be identified through gene sequencing, which can lead to cancer, and that can be identified before the cancer onsets. So this way they can get targeted treatment. And for those PCB water pipes that I mentioned earlier, people can check with their local city and county centers to see if their homes are exposed to those old water pipes, so you can prevent any sort of contaminants from getting into your water or your food and make sure that the paint in your house is also safe because PCBs also affect paint made pre-1979. So there are also some initiatives working to remove all of these pipes throughout the United States, so that's very important. And again, cow dairy is almost everywhere. It's in a lot of different products, so it's pretty much impossible to avoid it entirely. However, limiting your consumption of it and supporting groups that are trying to work to better filter microRNAs will help with long-term health. And another thing, there's a chemical called phthalates, which are a different type of chemical that is found in a lot of different beauty products, such as shampoos. And these have been found to mess with hormones and cause cancer as well. So it's very important to avoid those as well. So to conclude, answers to the question, how do genetic predispositions and environmental factors impact the onset of DLBCL? By permanently altering genetic code and removing p53 tumor regulation, by actively interfering with human signaling pathways like that IGF-1 signaling pathway and preventing apoptosis, and lastly, by bioaccumulating in human fat and tissue cells over continuous exposure to the carcinogen. So as for my experience, Dr. Prahlad and Dr. Virgil were very helpful with the process and really tried to ensure that my paper would be the best possible version of itself for publication. Dr. Prahlad was very helpful throughout our sessions and with the content of my paper. He gave a lot of very helpful comments on my paper, and he made sure that my paper was in depth and gave me many options to make my paper even more detailed and in depth. As for Professor Virgil, he made sure that my paper had the best wording and language to make sure that my research was more cohesive. So I'm very grateful for that. Thank you.
Very detailed and very informative. Kudos to you for putting in that kind of effort. I know our program is not easy. I mean, it's day in, day out. We use a coaching technique called immersion, where we teach a lot of things in a very short amount of time, and you're forced to flex the muscle called your brain. So it's been amazing. And I want to tell everybody to not give up, because we do have our research paper that needs to be submitted for publishing. We're just going to take a short break because you have your AP exams and everything. We're not going to bother you from May 1st to 16th. I believe that's when the AP exams get over. And I'm going to invite you back. I'm going to show you how to submit the paper. And please finish off your one-on-one sessions with Dr. Prahlad, with Professor Virgil. Get it done. Meet the deadline. I mean, just give me another month. May 31st, your research paper will be submitted. We would like to meet that deadline.
Same question, Dia. So what was the most intriguing question that hit you during the process, and how would you want to study further if you're given support?
So the reason why I chose this was because I had a family member who had DLBCL, so I would be very interested in studying it further. So I would be most specifically interested in gene sequencing and helping oncological scientists with discovering new cancer sequences. There are some specific mutations and codes, like I mentioned earlier, that scientists have already identified with DLBCL, and I would like to help with saving more lives and contributing to early detection in that way.
Great answer. Thank you.
Thank you.
Great job. Great job.